I'm super excited to introduce our next speaker, who is our keynote speaker, Dr. Dario Neri. Dr. Neri is a professor at ETH Zurich and serves as Chief Executive Officer and Chief Scientific Officer of Philogen, a Swiss-Italian biotech company which he has co-founded. Dr. Neri is a pioneer in the field of DELs, as he is one of the early designers and developers of DELs. He has published more than 400 articles in peer-reviewed scientific journals and is the recipient of many prestigious awards. His group and Philogen focuses on the engineering of therapeutic antibodies for the therapy of cancer and other angiogenesis-related disorders, and on the development of DNA-encoded chemical libraries, of course. Thank you so much, Dario, for your willingness to be here today and giving us the keynote talk. So Dario's talk is entitled DNA-Encoded Chemical Libraries from the Bench to the Clinic. Dario, the stage is yours.
Well, Sarin, many thanks for the very kind introduction. And I would like to thank also Anton, David, Tim, and the other co-organizers for the invitation. Let me make it full screen, and I'm ready to start. So it's a great pleasure, it's an honor. I will share information as honestly as I can. So I hope nobody is offended in any way by the statements I make. I think it's about truth and about research. And if I'm wrong in any way during my presentation, I apologize in advance. So the presentation is divided in three parts. First, I'll give my own, view on the history and key developments of the technology. I will then comment on few aspects which I think are very important, but sometimes not really spelled out. And then I will finish showing how we are using DEL technology for our research. DEL can be used for many purposes, but I will focus on one particular area, which is pharmacodelivery. I would like to start by really drawing your attention to the analogy of two very popular selection and amplification schemes. The first one to the left, antibody phage display technology, has been a big contributor to the Nobel Prize of George Smith and of my teacher, Sir Gregory Winter. And you see an antibody fragment binding to a protein, and you see that the phage contains the genetic information that codes for the antibodies. So this strange object embodies a phenotype, a binding phenotype, and the corresponding genetic information that allows us to amplify, those rare binding specificities which are fished out of a library. And in a way, DNA-encoded chemistry does the same. There is a small organic molecule that serves as binding phenotype, and the DNA fragment, it's not really biosynthetically coding for the molecule, but it serves as amplifiable identification barcode. Now, also the selections are very similar. With antibody libraries on phage, you typically screen more than 100 billion different clones. A few of them will bind to the protein. Actually, against virtually any protein, you get good antibodies with this technology. You wash away unwanted specificities, and then you elute those binders. And here you have the luxury of repeating rounds of panning if you need, or going straight to screening like ELISA. Now with DEL technology, in theory, you can screen more than 1 billion molecules. And I will be very critical about this statement with data. But anyway, you have big libraries, you can pan them. Some molecules will physically interact with the target protein of interest. Unwanted specificities can be washed away. And then, of course, PCR and sequencing are crucially important for discovering those rare molecules that may be fit for the purpose. And again, I'll speak about PCR and sequencing. Now, it all started with a paper that we all know by heart, really with the vision of the late Sidney Brenner, a very important Nobel laureate, and also of Richard Lerner, a pioneer in chemistry, and their vision of encoded combinatorial chemistry. Even though the paper in PNAS was basically a theoretical paper, it very lucidly spelled out what you could do on beads. So growing, for example, peptides and DNA with alternate synthesis. But also when we look at the patents that they filed, the authors were very clearly aware of many different ways of putting DNA together step by step, for example, by ligation. Now, this was 1992. There was a demonstration, or better, a couple of demonstrations with model peptides, but then the field remained silent for basically 12 years until a number of papers started to come out, and in particular, two groups actually showed that you can build libraries without beads, so attaching molecules directly to DNA. And already at that time, we started seeing different approaches. For example, David Liu pioneered DNA templated synthesis. So the idea that we would have DNA as preformed molecule to guide the synthesis with hybridization of oligonucleotide conjugates that would transfer building blocks onto a nascent molecule. And I will briefly speak about this technology in a split second. We published this concept of dual pharmacophore libraries, so attaching two sets of building blocks at the extremities of DNA, but later went on also to describe single pharmacophore libraries in which you attach molecules, no matter how complex, to single molecules to the DNA fragments. In reality, actually, we published maybe a few months earlier than David Liu, because we were out in May 2004 with what we call the encoded self-assembling chemical libraries. And really the vision was to have sub-libraries that could anneal and form chemical diversity in a combinatorial fashion. So if you have 1,000 blue molecules and 1,000 green molecules together, you would create 1 million combinations. Certain visions like this with triple-stranded DNA have not really been exemplified in practice, but the encoded libraries with two pharmacophores, either for de novo discovery or for affinity maturation, works, and I will provide examples. David Liu, I think with his DNA templated synthesis, he certainly created an avenue to generate functional microcycles, and he and his group have nicely exemplified the potential of this technology. I make some statements again reflecting my own understanding of the technology. I think in all the approaches we see something good and something that could be improved. I think the templated nature of the library synthesis enables in principle molecular evolution. This remains the attractiveness of the approach, that you would recover the DNA and you could drive evolution by changing, for example, the oligonucleotide conjugates for synthesis. But I think there are some challenges associated with the need to use pre-synthesized oligonucleotide compound derivatives rather than just chemicals that you can just buy and use off the shelf. And for this reason, I don't think DNA templated chemistry is heavily used nowadays. DNA recorded chemistry, and I will exemplify it with a slide, I think has been more frequently used. This is one of our first papers on this technology that we published in PNAS. And in this paper, we describe not only the synthesis of a single pharmacophore DEL library, but also the use of high throughput sequencing, which I think has been crucially important for the success of the technology. I think the advantage of stepwise procedures for synthesis without the need for pre-made DNA conjugates makes the technology very flexible. There are many reagents that you can buy, but I think this is the reciprocal aspect. This embodiment of the technology is hardly compatible with molecular evolution. In other words, if you want to evolve your compound, you have to build a new library. And I think, at least that's my understanding of the technology. And 1 year later, a very famous paper came out from GSK and Precis. So Barry Morgan was the senior author in this paper, and Barry and many other scientists are present today in this Congress. And I think the paper was important for many reasons. It described the synthesis and successful screening of a library with 800 million compounds. So really, the power of the technology was beautifully exemplified. And I think it's fair to say that similar research on DNA-recorded DEL synthesis was also going on at New Evolution, now Amgen, and possibly at other companies at the time. So I try to base my judgment on publications. This is a paper that I'm very proud of. Very early on, we actually used DEL technology to get affinity maturation of lousy binders. We often speak about DEL as a technology to discover hits, but actually DEL can be very powerful in affinity maturing compounds. And here is the example. We were starting from benzamidine, which is basically a 35 micromolar inhibitor against trypsin and other serine proteases. And by building a relatively small combinatorial libraries in which we add additional building blocks, we could identify very rapidly an inhibitor like this one that had a 3 nanomolar potency against trypsin and more than 1,000-fold worse potency against related serine proteases. So I think there are many ways of using DEL, but certainly affinity maturation applications can be important. Now, DNA is not just a barcode. DNA is the barcode because it's really the only structure that can be amplified. And so we can think about other encoding methods, but in the end, when we build large libraries, we need barcodes that can be amplified. And at this moment in time, DNA is the only amplifiable structure, or let's say the only directly amplifiable structure that we can think of. So we will be stuck with DNA as a barcode for a while, I think. And there have been articles in Science and in Nature that already 5 years ago have recognized that after the initial mistakes in practicing the technology was getting more and more mature. And it was becoming more and more popular, not only in industry, but also in academia, because it's a quite cheap technology. And so it's actually a sort of liberating feeling that you can build libraries without being a millionaire. Now, there are different ways of performing DNA-recorded DEL synthesis. We are particularly fond of encoding DELs with single-stranded DNA. And so we can discuss about the value of using single-stranded or double-stranded DNA. But if you want to use single-stranded DNA, one way to do it is you have building blocks coupled to oligonucleotides. So one building block for each distinctive oligonucleotide, which has a certain code. And then you can perform coupling, for example, using split and pool technologies. And of course, a lot of the concepts that were developed for traditional combinatorial chemistry come handy also for DEL. And once you have performed your chemistry, thanks to splint ligation, you can extend the code. And so in a way, we record each synthetic step by a ligation process. And this procedure can be iterated so that you add building blocks and you progressively grow the DNA. And again, single-stranded DNA offers certain advantages, and I'll come back to this in a split second. Now, this is an example of a paper that we published 3 years ago. And Zhou Li was the first author in the paper. This was a library of peptidic macrocycles with 35 million compounds. Each molecule is identified by 3 codes that correspond to the 3 sets of building blocks. So basically what you see in this graph is a cube. Each dot in this cube is a molecule. The 3 coordinates identify unambiguously the building blocks the molecule is made of, and the 4th dimension, so to speak, is the color, this color bar that basically gives us the relative frequency of each molecule in the library assessed by the number of sequence counts. And I will come back to this display and also to the information that we have from counting DNA sequences. But the beauty, at least for me, of the paper was that we were going after many different protein targets, and we were getting fingerprints against different protein targets. And some of these fingerprints were actually useful in practice, and I will show examples. But I like the idea you start from molecules which are more or less equally distributed in the library before the fishing expedition, and then we get ligands, including ligands against difficult targets like cytokines. Now, this is at least my historical introduction. Now I would like to focus on points which I think are important and not always really openly discussed in papers. So the first point is I described this cube of molecules that are all bluish and they all seem more or less equally distributed. But in reality, when you do a random sampling experiment of independent events, statisticians like Peter Bühlmann at ETH told us that actually a negative binomial distribution may be a good statistical way to describe the experiment. And indeed, we found for many libraries that the distribution of the density for different sequence counts could be well described by negative binomial distribution, and this would create also the basis for a statistically sound method to distinguish what is just variation due to sampling and what is a real hit that has a significant enrichment over background. Now, this is the theory. In practice, at least both at ETH and at Philogen, we find it very useful to perform something which is sort of obvious to many chemists and biologists, which is to just perform replicates of selection experiments. I don't see them very often published, but I think they're very important. And here we show an example of selections against PI3 kinase in a paper that Nicolas Pavalli and collaborators, so we have published this paper earlier on in Nature Chemistry, and you can see, for example, a beautiful reproducibility of the fingerprint. And if you focus, for example, on this brown dot, which is the most enriched compound, you see it clearly in 3 out of 3 independent selection experiments. So I would argue repetition of experiments is useful in this field. Another topic which is often discussed is once you see a fingerprint and you have trust that your enrichment factor is sufficiently large compared to noise, how do you validate hits? Of course, if you have an enzyme, you resynthesize the molecule and you use it in enzyme inhibition. But I think there are general methods which are by now very robust, and I would be an advocate for these methods. We like very much to resynthesize molecules as fluorescein conjugates because this enables in solution KD determination, for example, by fluorescence polarization. I apologize for the typo here. Also, if you have a fluorescent molecule, you can bind it to the protein on solid phase and you can detect fluorescein with antifluorescein antibodies. And in reality, the small molecule now becomes like an antibody. You can perform ELISA. The complexation of fluorescein with ligands such as antibodies allows you to also treat the small molecule like a protein, and then you can perform sensitive Biacore experiments. If you have enough protein, you can use isothermal titration calorimetry. We find it very useful with fluorescently labeled molecule also to bind the molecule to the protein, run it on a disposable PD10 column, and usually gel filtration gives you a realistic demonstration that you have sufficient kinetic stability for the complex to go through the column as a complex. Now, I mentioned affinity maturation, and I think affinity maturation is a big opportunity both for single and for dual pharmacophore libraries. With single pharmacophore library, if we have a molecule consisting of 2 or more building blocks, then the information that we get from the selection and also structure activity relationships may inspire the construction of analogs or even the construction of affinity maturation libraries. With dual pharmacophore libraries, you discover fragments which may be synergistic in interacting with the target of interest. And the linker, as you can imagine, plays an important role. First of all, it should not be too long and too floppy, but we have published examples in which linkers with different distance between the big building blocks, but also different rigidity, may give you, affinity increases, and in some cases you can even worsen the affinity. So there is a component of trial and error in the choice of the linker. Now, this is a paper which I'm very proud of, and I think it touches an important topic which we should discuss more often. So in this paper, we try to quantify, first of all, how efficient is the capture procedure in selections. The experiment was so performed, you would have a number of carbonic anhydrase binders tagged with DNA, and we had, for example, acetazolamide, which is a 10 nanomolar binder, this phenylsulfonamide, which is roughly 100 nanomolar binder, and this metasaba, we call it, which is a 1000 nanomolar binder. And then you can perform capture experiments on solid phase, on carbonic anhydrase 9, you can use negative controls, and you can use qPCR quantification. Of course, if you want to use qPCR quantification, you have to start with a unrealistically high number of copies, so typically 10 to the 11. And so you can look at if you want your positive control or your negative control, you can see how many copies you have before selection, how many copies you have after selection, what happens if you have a no protein control, what is the PCR background. And in short, we have published in a number of papers that in our hands, the capture efficiency at these doses of DNA can be anything between 10 and 90%. So the capture can be efficient, but you will see that it's not only capture, it's also the PCR efficiency that contributes to the size of the library that you can handle. And for example, if you look here at this figure from the same publication, you look at del selection against the carbonic anhydrase, and basically these fingerprints correspond to different inputs of the library, and you can measure them like this. You can say, okay, each library member was present in 10 to the 8, 10 to the 7, 10 to the 6, 10 to the 5 copies, or you can look at a row, at a stripe, and you can say, okay, collectively here you had 246 times the same good building blocks. So it's a matter of taste whether you like to see this as an experiment in which you look at the individual dot or you look at the cumulative line. But I hope you can see that at least in our hands, when we go below a certain threshold, be it 10 to the 4 in our hands, it's more often 10 to the 6. The capture and the PCR amplification is not efficient enough to give you reliable binders. So in our lab, we use at least 10 to the 6, preferably 10 to the 7 or more copies of each compound to have reproducible selections. If other groups are more efficient and better, I would love to see this data published, because then we can really compare methodologies and protocols. There is an obvious implication that if the selections are not terribly efficient, then the construction of very large library becomes useless because we cannot detect reliably the binding events. So I think it's an important point, at least in my opinion. Let me also mention that the selections against cells are not always easy to perform, and we have recently published again our quantification on the efficiency of those procedures. Now, I mentioned that single-stranded libraries are versatile. We like them because you can use them as such for affinity capture followed by PCR and sequencing, but they enable also the use of complementary strands equipped with a photoreactive group so that you can do selections by photocross-linking, and this was pioneered by Xiaoyu Li at University of Hong Kong. Or you may use libraries that self-assemble and create larger repertoires, either as dual pharmacophore formats or even what we call ESAC plus when you create cyclic structures. And I'm sure we will see more and more examples of how you can assemble chemical moieties on DNA. Now, in the last part of my presentation, I would like to share with you at least the main application for which we use DEL, and I think that the technology is really transformational for certain pharmaceutical applications. So we like to use DEL technology mainly to deliver drugs and radionuclides to the site of disease. So this is exemplified in this slide, in which you see a validated ligand against a protein called FAP, fibroblast activation protein, and I'll show you the work in a split second, but we have applied similar concepts for carbonic anhydrase 9, for placental alkaline phosphatase, and for other targets. And so in a way, I think what you see in this slide is representative of a platform rather than a one-off story. And the idea is if you have a small molecule that efficiently targets tumors, in this case, then you could use it to deliver radionuclides for imaging or therapy. Or you can really compete with antibody drug conjugates, creating small molecule drug conjugates, which are inexpensive to produce and very well performing. And I'll show you data, how you can really equip these targeting moieties with linker payloads. You can also start to dream and compete with antibodies also in the field of bispecific antibodies, which are so popular nowadays. So rather than having two antibodies that cross-link a tumor and, let's say, a T cell, you could have two small organic molecules that bridge together those cells in order to enable active killing. Or we have described also the use of small organic ligands equipped with fluorescein to redirect so-called universal CAR T cells against the tumor. If you have CAR Ts equipped with an anti-fluorescein antibody, then this bridge will link the universal CAR T with the tumor cell. And for example, Powell and also Phil Lau have published beautiful work in this area before us. Now, why would we bother to do pharmacodelivery? Well, in cancer therapy, unfortunately, most conventional cancer therapeutics go everywhere except to the tumor. So you have a poison that is distributed in the body but doesn't really localize where you need it. This is work of a group in Amsterdam at the Free University Medical Center. What the colleagues did, they took docetaxel, which is a poison, tubulin poison, a frequently used chemotherapy. They labeled it with carbon-11. They gave it to a patient with mesothelioma. But you don't see the tumor in the pleura. You see anything else. At early time points, you see the blood pool, the main vessels, the liver, the spleen. Then the clearance continues. You start to target the bone marrow. At later time point, the drug is out in the feces and in the urine, but at no moment in time, the tumor is visible. And this is not an unlucky drug and an unlucky patient. This is a characteristic feature of chemotherapy. So you can make it better. For example, if you take a small molecule which is fluorescent in the near infrared field, it works like chemotherapy. It goes everywhere except to the tumor. You give it to tumor-bearing mice, and you see first you have distribution in the body, then the molecule goes out through the kidney, and at no moment in time you see an accumulation in the tumor. You couple this payload, in this case to acetazolamide, which is a ligand to carbonic anhydrase 9, a marker of renal cell carcinoma, and now this is fluorescent in a matter of 1 hour, and it stays fluorescent for a long time. Eventually it falls off because the dissociation constant is only 8 nanomolar, but if you improve the affinity thanks to DNA-encoded chemistry, you make a molecule with an affinity in the picomolar range, and this time not only you get to the tumor in 1 hour, but the molecule stays longer to enable, as you will see, a selective therapy. Now, importantly, this principle has now been validated, not only in mice, but also in patients. And so we have recently published in collaboration with colleagues in Vienna, the demonstration that the small molecule can also localize. For example, here you see a patient with lung metastasis, lymph node metastasis, and you can see that the molecule preferentially localizes to the site of disease. Having ligands to tumors allowed us also to perform the first side-by-side comparison of antibody drug conjugates and small molecule drug conjugates. And you see in this case, an antibody and a small molecule against carbonic anhydrase 9 equipped with the linker payload of Adcetris, so a cleavable linker and monomethyl auristatin E as a payload. And what we learned is what we feared, that the ADC, at least in our hands and in other publications, only localizes to perivascular sites in the tumor. It gives you a patchy uptake, whereas the small molecule, it extravasates more rapidly and targets better. If you use binders of irrelevant specificity, then this green color disappears because the molecules without a specific binding do not localize to the tumor. And when you look at therapy, so you're looking at tumor size versus time, you can see that actually both the small molecule drug conjugates and the ADC can be active against the tumor, but while the ADC in our hands gives not a fantastic discrimination between specific antibody and non-specific antibody, with the small molecule drug conjugate in our hands, we get better results. So good that we start getting complete cures in tumor-bearing animals. And one of the nice features is that the targeted delivery of cytotoxics exemplified by these molecules can be potentiated by the delivery of cytokines. And this is a feature that we like to exploit. It's nicely seen in this picture when you hit the tumor with a targeted cytotoxic and a cytokine, you turn a live mass basically into a hematoma, which is heavily infiltrated by natural killer cells, and tumor cells are dying. Now, I finish with 2 quick examples and then I shut up to actually show you where the field is going. Fibroblast activation protein has been dubbed the next billion-dollar target, if anything, because of the beautiful work of Uwe Haberkorn in Heidelberg. What he has done with ligands against FAP is to basically target many, many different tumors in patients. Here you see PET images. Look, for example, at this colorectal cancer patient. You see the liver mets and the lung mets with extreme precision. And if you contrast this image with the one that you get with an antibody, you get the feeling that chemistry can be better than antibodies for targeted delivery. Uwe Haberkorn and collaborators have focused on a chemistry for the modification of the quinone ring. We found that position 8 is particularly suitable for giving you high affinities. And we have recently published, and this is the leadership of Samuele Cazzamalli within Philogen in this PNAS paper, that we can get tumor to organ ratios which are sensational already 10 minutes after injection of our ligand. And as time goes by, the selectivity gets even better. and the targeting is stable until you reach a dose, in this case, 1 micromole per kg, at which you are saturating the target in vivo. So you can imagine that these results are beautiful in mice, but we have images in patients which are about to be published, which are the most beautiful pictures I've seen in my life. And so I think this gives us the motivation to go forward. And the last 3 slides are only to show you a third example, again, to exemplify the potential of chemistry for targeted delivery. This is the work, again, of Samuele Cazzamalli, Gabriele Bassi, Nicola Spavalli. You see PLAP, placental alkaline phosphatase, which is a very good marker for gynecological tumors and also for testis cancer. The only problem until now, small ligands against PLAP were not known. And it is challenging because there is a closely related target called TNAP, tissue non-specific alkaline phosphatase. So you want to get not only a good PLAP binder, but also a specific binder that doesn't bind to TNAP. And to cut a long story short, we performed selections with many different libraries, and we found selective PLAP inhibitors in, for the sake of time, I will not comment this slide so much, but rather the next one that shows the structure of different hits that were identified. We looked at different stereoisomers, and we look at IC50 in inhibiting basically PLAP. This molecule to the right, constructed thanks to Suzuki chemistry, is a potent, so it's a nanomolar inhibitor of PLAP, which basically does not bind at all to TNAP. And when you equip this structure, which is repeated here with fluorescein, you can do the beautiful things that I've shown you before. It works like an antibody. It will stain by FACS antigen positive cells, but it will not stain antigen negative cells. You can see again by fluorescence microscopy how the small organic molecule works like an antibody on the positive cells, but not on the negative cells. And so to cut a long story short, I think DEL is helping also the discovery of ligands that may facilitate pharmacodelivery. So I believe that DEL technology, thanks to the effort of the community which is present today, has become popular for the discovery of hits, but also for affinity maturation. I think DEL allows the construction and screening of very large libraries, but I don't think it's still formally demonstrated whether large libraries can efficiently be screened, especially if you don't have efficient selection and PCR methods. In our experience, it is good practice to have at least 1 million copies for each compound, preferably 10 million copies. And at least our own work shows that ligands specific to good quality tumor antigens may revolutionize the way we treat cancer because they enable selective targeting. This is the fruit of the work of many talented persons that are listed in the slide. I particularly would like to mention the long and fruitful collaboration with Jörg Scheuermann at ETH Zurich, whereas within the Philogen group, small molecule therapeutics are led by Samuele, and I have mentioned the work of the talented scientists listed in this slide. With this, I would like to thank you again for the honor to give this lecture. I shut up and I'm glad to take questions if there are any.
Thank you so much, Dario, for this fantastic talk. Yes, there are questions. So I'm going to go ahead and ask the first one. So first question is, what in your experience allows single-stranded DNA DELs to provide better coverage of chemical space compared to double-stranded DNA DELs?
No, I think the chemical space, when you use them as single pharmacophore libraries, they're basically equivalent. And we have actually published a comparison of single-stranded versus double-stranded DNA, and the performance is basically identical. The only difference is that when you have the library on single-stranded DNA, you can use it as such, but you can do additional things. So you can pair it to another strand to play ESAC technology, or you may use photocross linkers to enable different selection modalities. So all I'm saying is that in our hands, the chemistry has a similar performance, but the library is more versatile. If scientists are happy with double-stranded DNA, they should stick to double-stranded DNA, which works as well.
Thank you, Dario. One more question here. Can you comment on the difference between, okay, so I think we just answered that, between single-stranded and double-stranded DELs in regards to selecting against DNA binding proteins?
Yeah, so that's a good point, because you will have DNA binding proteins that bind to double-stranded DNA. And so I think chances are that in that case, single-stranded DNA will not be the obvious target for a molecule, for a protein which is designed to bind double-stranded DNA. The opposite is also true. If you have a molecule that recognizes single-stranded DNA, then you may want to use double-stranded DNA. The beauty is that once you have it single-stranded, you just do Klenov polymerization and you convert it into double-stranded DNA. So in our experience, we see only plus in starting at least with single-stranded DNA. But in German, you say it's Geschmackssache, it's a matter of taste, and so people will choose what they like best.
Thank you, Dario. There's multiple questions coming in, and we have a few more minutes. So, so the next question is, what was the validation rate for the hits that you got from screening? That's the question.
I think the validation rate, if you are honest about the fingerprints, is very high, meaning, in my opinion, certainly above 80%, but you have to be honest about the fingerprints. I think, honestly, we have made mistakes in the past taking enrichment factors that were too low, okay, and not repeating the experiments. The moment we started using a lot of copies, performing experiments, at least in duplicate, better in triplicate, and really performing a rigorous analysis of enrichment factors using libraries which are pure, then you are really not fooled by the technology. And so I think I would not agree 100% with the statement of Tim before that technology, doesn't deliver too many hits and may fool users. I think it crucially depends, in my opinion, on how you practice the technology. And also the opposite is true. If your fingerprint is ugly, I think it's okay. The technology didn't work. It was not good enough for the purpose. I think as a community, we publish much too little about failures. We try to be honest with the data, but I think the day that, also journals encourage people to publish success and failure, I think the whole community will benefit.
Wonderful, thank you. There's many more questions coming in, so we're gonna take a few more minutes and answer questions. Your talk was really exciting. So one question is, so thank you, Professor Dario. It was an interesting talk. I know the PET radionucleotide has false positive results. How would you control that with DEL?
Well, PET technology, in my opinion, is one of the most accurate and honest techniques you can think of, because the radioactivity doesn't lie. If it goes to the tumor, you see it. If it doesn't go to the tumor, you also see it. So I actually, if I had to name one technology that I would blindly trust, is PET radiopharmaceuticals, and that crucially depends on the quality of the ligand. So it's not the only area in which DEL is useful, but DEL is really transformational in this field. So this localization is not a coincidence. That's what I wanted to say. And we have formally proven actually that affinity, binding affinity, drives performance. So the better the ligand, the better the performance.
Fantastic, thank you, Dario. I know some of our panelists had questions for you. So I believe, Tim, you had a question? Yeah, go ahead, Tim.
Yeah, hi, Dario. I had a question about the percent efficiency of selection that you observed. I think that's an interesting observation and one that's been made by multiple groups in the field. I'm curious if you have any thoughts about why that is and how that applies to thinking about the upper limit of, or the weaker limit of potency based on what you could discover from this apparent low recovery.
Yes, at least in my experience, it's very difficult to generalize it, and you know better than me. But I think it's fair, because everything depends on how you perform the affinity capture. But I think it's fair to say that if you have a KD which is 1 micromolar or better, usually you see it. And there are plenty of model experiments that one can repeat, including the ones with carbonic anhydrase or other binders. So I would say if the library contains a 1 micromolar binder or better, this will shine. But the second aspect, and I'm very passionate about it, and people may disagree, is you need to know your own PCR amplification and selection conditions. In our hands, we need so and so many copies. If you are better, we would love to read your method and use it, but I think that is the interplay. I think the two conditions is you need a binder which is at least 1 micromolar, and you need a sufficient amount of that binder. Everything else is straightforward, in my opinion.
I agree with you. Thank you.
Thank you. Dr. Neri, you still have a few questions. If you don't mind, maybe looking at them after your talk. Thank you so much for your time, this exciting presentation, and taking time with all the questions. So appreciate it. Thank you so much.
Thanks a lot. And I'll reply in the Q&A. Thanks a lot.
Fantastic.